My name is Makie Matsumoto-Hervol, and I am a 2013 JISAO intern. I partook in a non-invasive study by the University of Washington's Center for Conservation Biology to collect and analyze southern resident killer whale scat. These whales, declared endangered in 2005, are threatened by three probable factors: dwindling Chinook salmon numbers, toxic pollutants in the water, and vessel traffic. In late June, I traveled to San Juan Island and became a part of an amazing field team. On a 21-foot motorboat named Moja, we scoured the Salish Sea, tracking whales and their scat. The driver, Carrie or Giles, evaluates the wind and current of the water to maintain a 200 to 400 meter distance from the whales. Meanwhile, two research technicians, Amanda, Sarah, and I, snap whale ID photos, communicate the movement and proximity of the whales, and record vessel numbers and whale behavior data. When a conservation canine is downwind of a scent, they indicate scat detection by actively moving around the edges of the bow, sometimes barking or whining loudly if the scent is close. Based on the sea canine's behavior, the sea canine handler Liz would communicate with the driver to maneuver towards the orca poop location. When the scat is spotted, the team uses plastic beakers to quickly but delicately scoop the scat, careful to prevent it from sinking or dispersing. The scat pieces distributed among the beakers are then compiled into a 50 milliliter tube using a sterilized pipette to coax scat movement. The sea canine is rewarded with playtime for their success, while the sample's size, color, texture, and other descriptions are recorded. The sample is then centrifuged on board for 5 minutes, resulting in often 10 milliliters or less of whale scat. It is then immediately frozen on board, destined for travel to the University of Washington's Wasser Lab. In August, I joined my mentor, Jessica Lundeen, an environmental toxicology grad student, and assisted with preparation for whale scat hormone, toxicant, and microbial analysis. We first thawed the frozen samples, and in a much larger centrifuge, re-centrifuged them for 20 minutes to further condense the scat and separate it from the seawater. The samples were then reassessed of their field descriptions and swabbed for DNA analysis conducted by NOAA. Though all samples are homogenized, I also spent time homogenizing large samples in multiple tubes by combining and mixing them in a beaker. After many detailed notes, icky fish smells, and thorough labeling, the samples were frozen for 48 hours for freeze drying. When ready, the samples were placed in a freeze dryer, which pressure vacuumed the remaining water out of the samples, leaving only powdered whale scat behind. Through my internship, I created unforgettable experiences immersed in amazing communities on San Juan Island and Seattle. Thank you for an amazing summer adventure.